All right. So, good morning, everyone. Good evening, everyone. And thank you for coming for our second virtual series of summer workshop, Microscopy 101, that co-hosted by BiPS and BBUGS. This virtual series comes with 3 sessions, and today is about Introduction to Microscopy for Biological Science. Briefly, I will introduce BiPS. BiPS is an acronym of Biomedical Photonics Society, having a vision of serving as a platform for graduate students working in biomedical photonics and anyone who are interested or microscopy users. We are an officially chartered organization at Georgia Tech, and moreover, we are an official student chapter and sponsored by OSA. The purpose of this workshop is to provide simplified fundamental knowledge on microscopy system so that all attendants could bring and gain better understanding on recent microscopy systems. The current workshop is recorded and will be uploaded in the homepage. Today's session is about 45 minutes and Q&A session will be following. Today's subtitles are Parameters of microscopy, light sources, illumination and detector, and microscopy systems. All right. Before starting with the parameters of microscopy, let's back our memory to the last session. So we will upload last session video in the homepage later soon. But once again, with 2 lenses called 4F system, we can build a fixed magnification system, calculated with the ratio of the focal lengths of 2 lenses. Basically, microscopies also follow the same rule, even with the complicated recent microscopy as well. For microscopies, lens 1 will be objective lens with diverse focal lengths upon your choice, and lens 2 is tube lens that is located inside the commercialized microscopy system. There are parameters to decide the performance of microscopy system. We will go over today one by one at this time. Diverse lives has different scales of size, but imaging system has physical limitation. With light microscopy, the smallest size of target can be only achieved down to 200 nanometer, except super resolution microscopy techniques that we will deal with next session. Then what decides the limitation of microscopy, especially resolution? Let's start with the lateral resolution, meaning XY plane. When you throw a ball on a pond, it generates water ripples. It is physical phenomenon diffraction. With a small aperture, light also generates diffraction pattern. The energy graph shows 1D diffraction pattern. With microscopy, the infinitesimal light source diverges through an objective lens and propagating. With tube lens, which is another 3D aperture, the light is converging to generate 3D diffraction pattern called Airy disk or point spread function. Literally, it means the function of how point spread through microscopy system. When projecting it in 1D, this point spread function shows a sinusoidal pattern. Okay, we go over the case for single source here. What about 2 sources? When 2 light sources are close to each other, microscopy cannot differentiate it as 2. At certain distance, which is the smallest resolvable distance, 2 sources can be resolved as 2. The distance is calculated by Abbe's resolution criteria. Interestingly, the 0 of red graph is at the center of the green graph, so that distance is identical to each fluid top maximum in this criteria. Now, what is an A? Na is the ability of objective lens to gather light, and it is a function of refractive index and converging angle. The sine term can be approximated as tangent, so that it is now the function of diameter of lens and working distance, which is the distance for image to be focused. In result, higher Na have higher angle and good performance, but it is more expensive. With the same diameter, higher Na has shorter working distance, meaning we need to place sample more closer to objective lens, vice versa. Second parameter of microscopy is depth of focus. With a photography, for example, a narrow depth of focus limits the achievable depth of the image, so that we can clearly image within only certain distance. With higher depth of focus, we can image both objects placed closer and farther as well. Let's back to microscopy for depth of focus. In microscopy, depth of focus is also called axial resolution. After objective lens, light has focus beam and two tails up and down. With lower Na, these tails are elongated than higher Na, meaning you will have higher depth of focus. In depth of focus equation, it has the square of Na term in each denominator. Unlike photography, where higher depth of focus is better to have object closer and object farther at the same time, in microscopy system, lower depth of focus are better for higher SNR, signal to noise ratio. Because high depth of focus meaning you will have unwanted background from out of focus plane. It is also called optical sectioning ability. Next parameter is field of view. Field of view is the size of region where microscopy can image. This field of view can be decided with 4 parameters. 1. Magnification. 2. Numerical aperture. 3. Diameter of lens, which is typically 1 inch in commercialized setup. 4. The dimension of camera chip. With higher magnification, you can get higher zoomed image, meaning field of view is decreased. Next is numerical aperture again. With fixed diameter, the contribution of Na to field of view mostly depends on magnification, and higher Na objective lens has a higher magnification. Then, how can we calculate field of view of system? Microscopy system. Then we need to talk about a dimension of camera chip. First, let's define 2 planes called as an objective plane and image plane. In an image plane, field of view is magnified, so we do need to consider this magnification factor, because the image is magnified and divided by sensor chip size itself. For example, first we need to calculate the effective pixel size, that is dividing sensor size by magnification. Typical SCMOS sensor size is 6 micrometer with 100 magnification. Now it is 60 nanometer of effective pixel size. Next, field of view is the area of the product of the number of sensors and effective pixel size. So that with the number of sensor size in SCMOS again, final field of view can be calculated to about 70 nanometer by 40 nanometer. 70 micrometer by 40 micrometer. Finally, what about objective lens of microscopy? Inside an objective lens, there are multiple of lenses acting like a single lens. Then how do we differentiate objective lens among others? Simply, most information are written on the cover of the objective lens itself. Magnification, numerical aperture, and emergent media, etc. Some objective lenses work in air, water, or oil immersion status. Depends on your applications, we can choose different immersion objective lens. It is because these objective lenses are designed to increase acceptance angle, which is NA, so that maximum NA for resolution. Typically, oil immersion lenses has higher NA than water or air objective lens. With microscopy system, we face aberrations, which is artifacts from multiple regions. The most common aberration is chromatic aberration, meaning different wavelengths, different color of laser or light source has different reflective angles, so that different color of light has slightly different focal plane after objective lens. For example, different color images of same sample are located in slightly shifted position in your image. The chromatic aberration happens in 3D dimension, so displacing focal points of different colors. How could we solve it? Commonly, apochromatic objective lens can be used, so most of commercialized microscopy systems equip of this system. But for high or super resolution microscopy cases, this is not enough. So that it is required to generate calibration data for post-processing after imaging with different colors to replace all at the same place. So in the bottom images, after collection, 3 color images are well overlapped in same position. Second most aberration is spherical aberration, which is from relatively large curvature of the lens bring non-identical focal plane. With ideal case without aberration, 3D point spread function is symmetric. But for spherical aberration system, there are large curvature at the side of the lens than compared to the center position. So that light are focused at different position and generate asymmetric point spread function. And also the edges of images get blurred, so that effective field of view decrease. Spherical aberration increases with larger diameter of the lens. The simple solution is by limiting the region of sphere surface, but practically, a U-plan objective lens is used to correct this correct this spherical aberration. Yes, so now we have gone through parameters that decide the performance of microscopy. Next is Nishita to talk about light source, illumination, and detector.
Hey everyone. So now that you all heard a little bit about objective lenses and some of the important imaging parameters that are controlled by them, such as the resolution, the depth of field, and the field of view, let's talk about another aspect of imaging which is equally important. And this is essentially what allows us to form the image. And this would be just the source of light or the illumination. So to start off, let's take a quick look at some of the light sources that are used in microscopy. So you might wonder what parameters need to be considered while choosing a light source. And the first among these is just the brightness, which basically means how intense a light source is. So this depends not only on the source, but also on the numerical aperture of lenses in the system. Like we talked about before, numerical aperture essentially limits the range of angles that can be of light that can be accepted by a system. So having a larger numerical aperture means that you can accept more light and therefore you have higher brightness. Another important factor is basically the bandwidth, which refers to the range of wavelengths or frequencies that are emitted or given out by a light source. So in the image on the right, you can see something that's known as a spectral profile or an emission spectrum of different types of light sources. So for example, you see the blue line that corresponds to the tungsten halogen lamp. And this is basically a pretty broadband source in the sense that it has multiple wavelengths that are being emitted at varying intensities, and the variation in the intensity is also pretty smooth. In contrast, if you look at a mercury or a metal halide lamp, these do have quite a big range of wavelengths or frequencies, but you tend to see distinct peaks at specific wavelengths. Now, depending on the sample that you're imaging and the type of contrast mechanism you have in place, you might want to choose different types of sources for different applications. Another thing to consider is uniformity across the field of view, which just means that you want to have uniform illumination across your sample, and this is generally the case for most types of microscopy applications. Finally, another important concept is that of coherence. And in very simple terms, coherence just refers to self-similarity in light waves or wavefronts, both in space and in time. So spatial coherence in simple terms just means how similar light waves or wavefronts are to each other at different points in the transverse direction of propagation of a wave at a particular instant of time. And temporal coherence refers to how similar a wave would be to a delayed version of itself. And this is basically very related to how narrow the bandwidth of the source is, or in other words, how monochromatic you can consider it to be. So the closer it is to emitting just a single wavelength of light, the higher the temporal coherence. So as you can see in the image at the bottom, going from the top left corner, you have light of varying wavelengths indicated by the different colors. And you also see that they all look very random and there's no specific relation between them. This basically indicates spatial and temporal incoherence. And then finally, on the bottom right, we have light of a single wavelength, and all the waves look very uniform, and there's a very nice relationship between each of them. And so this refers to both spatial and temporal coherence. Now, for most microscopy applications, we can actually get away with using just spatial as well as temporally incoherent sources. Okay, so now that we have a sense of these different features, let's look at some example sources that are actually used. So the first type of sources that you can think of are broadband sources. So these are also called polychromatic because essentially you have a range of wavelengths. Or if you're looking at the visible region of the spectrum, you could also call these white light sources. Now the simplest and the most readily available natural source is the sun. And this is actually a very broadband source of light. And this is what the early microscopists used to be able to emit samples. More commonly, we have incandescent lamps, which are basically tungsten lamps that are very commonly used for bright field microscopy, as well as a range of label-free techniques. And these are very readily available, but they may not be bright enough for applications such as fluorescence microscopy. Also, you need to be careful with these because they tend to heat up a fair bit. So at the other end, you have a narrowband source, or in other words, a monochromatic source. And the best example of these are lasers, which I'm sure all of you have heard of. These are extremely stable, extremely coherent sources. And while they are useful for a lot of applications, they're also extremely expensive and can be difficult to set up. So you don't always want to use them unless you absolutely need them. Now, a good medium between these two is arc lamps, and these refer to essentially lamps like the mercury arc lamp that we saw in the spectrum previously. So these have a fairly broad band of wavelengths or frequencies, but because they have extremely high intensities at specific peaks, we can use a filter and then they would behave as an effectively monochromatic or narrowband source. Of late, though, these have fallen out of favor because they tend to need to be replaced pretty frequently, and also they're often made of hazardous materials, so disposal is an issue. Finally, especially in the recent times, LEDs have become extremely popular for a wide variety of different applications. This is because they're very easy to produce, they're cheap, they're readily available, and they're extremely diverse in the sense that you can have very broadband white light LEDs, or you could have narrowband LEDs corresponding to very specific wavelengths, as you can see in the images of the spectra. And especially with improvements in technology, the LEDs have become very bright, and we are able to use them for all types of applications. Okay, so now that we have a sense of these different example sources, let's actually see what we need to do to be able to relay light from these sources or lamps to actually illuminate our sample. And so in order to answer the question of how our sample is being illuminated, it's useful to review some of the important concepts of image formation in a microscope. So essentially, you would want to treat a microscope as being made up of lenses and diaphragms. So what are these? A diaphragm is essentially just an opening that's adjustable and controls the amount of light. So as you can see in the image, you can either make it narrower or wider and adjust the width of the beam. Similarly, a lens, as we talked about before, is something that can either focus light or collimate light, and you can also form images depending on where the object is placed. So usually, if an object is placed at the focal point of a lens, this will generate a parallel beam of light. And this also works the other way around in the sense that a parallel beam of light will be focused to the focal point. And also, if you have an object outside the focal length of the lens, then you'll get an inverted magnified image in most cases, or the size can be different, but you end up with an inverted image. The important thing here to remember is that lenses can multitask, so that if you have one object at the focal plane and one outside of it, the first one would generate a parallel beam and the second one would produce an image through the lens. So let's look at another important concept, which is the concept of conjugate image planes. And this is not particularly hard to understand. It just refers to a series of planes where the images of an object are in focus. So on the left, you see a pink dot that's indicated by the arrow, and then you have a series of 3 lenses and a set of 3 conjugate image planes corresponding to that. Now, if we had 2 objects, we would essentially be able to see 2 sets of conjugate image planes corresponding to these 2. So we have one set corresponding to the pink or purple object and one set corresponding to the green. Another interesting feature of conjugate image planes is that if you were to place an object at the conjugate image plane of another object, then at every subsequent plane you would see these 2 images as being overlaid. So this is essentially equivalent to placing the 2 objects one on top of the other at the sample plane. And this is something that's frequently exploited in microscopy. Okay, so now that we have some sense of the basics, you might wonder why it's important to care about how we're setting up the illumination, or why it's important that everything's correctly set up. And this is very well illustrated in these example images. So on the left, you have a label-free image of cheek cells. And as you can see, the bottom right is clearly much darker, and in general, it's hard to distinguish features in the image, and it's not a good-looking image. And on the right, you see the image looks dramatically better, and everything is uniformly illuminated. And the only difference between the 2 images is just the alignment of the illumination optics, so that you have a uniform illumination at the sample. So let's get a better sense of how to achieve this. So this is usually achieved in most commercial microscopes by a method known as Koehler Illumination. This is used mostly for any bright field or wide field microscopy, as well as a majority of label-free techniques. So we already talked about the image forming optics being the tube lens and the objective, and you can have an eyepiece that relays the image to the eye, or you can have any type of image sensor at the intermediate image plane. But now, in addition, in order to relay light from the light source, we have these 2 new lenses called the collector and the condenser lens, and 2 diaphragms known as the field diaphragm and the condenser aperture diaphragm. Now, the field diaphragm, just as the name indicates, essentially is used to control the field of view of the image, and the condenser aperture diaphragm can just control the angle of light entering the condenser, so it can affect the numerical aperture of the system. So let's now look at the illumination conjugate planes to get a better sense of what's happening. So the way things are set up, if this were a well-aligned system, the light source and the field lens essentially are positioned such that an image of the light source is formed at the front focal plane of the condenser, where the condenser aperture is also located. So just based on the basic rules for image formation that we learned, if an object is placed at the focal point of a lens, then this generates a collimated beam. And so you have a collimated beam illuminating the sample, and you have an image of the light source being formed at conjugate image planes at the back focal plane, and at some plane that's before the sensor, so that at the sensor you don't have any image being formed. And this is important because if you had an image of the source, this would lead to sort of a non-uniform illumination. Another thing that we also need to look at in order to make sure everything is correctly set up is known as the imaging conjugate planes. So these would essentially, can you go to the next slide, please? Yeah. So these would essentially be the planes that we're already familiar with, such as the sample plane, the image plane, and finally the image on the sensor. But in addition, here we also see an image of the field diaphragm being formed at the specimen plane. And this is a really important feature that's exploited to actually be able to align everything up in a real microscope and be able to achieve a good illumination. So let's put these two things together and sort of see what happens. So moving on, yeah, so in a correctly aligned microscope, this is sort of what you would see with the imaging conjugate planes as well as the illumination conjugate planes overlaid over each other. So on the right, what you can see is if your objective is focused on the sample, you would see maybe an image that looks like the one on the top, where you can see an in-focus sample. And if you would close down this field diaphragm, you would be able to see an in-focus image of it that's well centered. So let's take a look at what this would look like in an actual microscope and what you might need to do to be able to set up correct Koehler illumination. So on the left, you have an upright microscope. This would be similar, but just directions might be inverted if you have an inverted microscope, but essentially all these same rules would still hold. And you can see the field and the aperture diaphragm indicated by blue and green circles, and all the other lenses and the light path being indicated. And on the right, you essentially have a magnified version just showing the field lens and the field lens and the condenser and the corresponding diaphragms. So the important thing to remember is that most of the lenses in a microscope are fixed, and we don't really have any freedom to move them. The only two that are movable is the objective, because obviously you want to be able to focus your sample. And you can also move the condenser both in the XY direction as well as axially to ensure that your illumination is correctly set up. So as you can see in the image on the right, you generally have like screws to be able to move the condenser in the axial direction as well as to center it correctly in the field of view. And then you would have rings to open and close different diaphragms. And so essentially each time you change out your objective, you want to check that you have Koehler illumination correctly set up. So this is the basic principles of how to set it up. And if you follow these, you should be able to get well illuminated samples every single time. Okay, so now that we looked at different types of sources and how to set up a good illumination scheme, let's look at things on the other end of the imaging system, which are basically the detectors or just any place where an image can form. So let's look at what types of detectors we can have. So one of the simplest types of detectors is basically just the human eye, and this is what is most readily available. So a lot of early microscopes essentially used a lens known as an eyepiece to produce a magnified image on the eye. And one of the disadvantages of doing this is that while you can see the images really well, there's no way to record them. So early biologists and microscopists would essentially hand draw out samples that they saw under the microscope. For example, there's this famous image of neurons drawn by Ramon Cajal based on what he observed. So eyepieces, like objectives, are pretty complicated lenses, but all you need to remember is that they essentially just form a magnified image of the image at the intermediate image plane on your eye, and these would affect the overall magnification as well as the field of view. So always remember that if you're using an eyepiece, then your total magnification is given not just by the objective, but the product of the objective magnification and whatever is introduced by the eyepiece. Now, detectors that have become much more common recently are basically electronic detectors. And these are really cool because you actually are able to record and capture and store a digital image that you can do a lot of further analysis on. And this is most commonly seen as some type of camera that's attached to a computer so that you can immediately store the images and process them on the computer. And for digital detectors, you might want to consider different factors such as how efficient and how fast they are or how you're sampling. And we'll get into that in a little bit, but first, let's really understand how all these electronic detectors work. So most electronic detectors essentially work using the same basic principles. So in any detector, you first have photons or light essentially hitting a photosensitive material. And through the photoelectric effect that we talked about in the previous session, if you all remember, we have these light photons being converted into electrons. And the efficiency of this process is something that's very important because obviously you want to capture most of the light that hits your detector. So you want to have a high efficiency or quantum efficiency, which is commonly listed as a parameter for many detectors. So once you have electrons, you can essentially generate a voltage or an electric current. And the voltage would be an analog signal, but you have an analog to digital converter to convert it to a digital signal. So as you can see here, there's about 8 different digital levels, but depending on the type of signal, you can have something like 1024 or maybe 256, 512. And this is essentially known as the bit depth or the number of bits you need to be able to store this digitized and discretized voltage. Okay, so now that we have some sense of how the detectors work, let's look at the two common types of detectors. And broadly, you can think about detectors in terms of being single point detectors, which basically means that they only capture a single point and relay that point to the detector. And because you're only imaging a single point from the sample, you essentially need to scan the sample. And you could do this either by moving the sample itself or by moving your light beam to be able to actually reconstruct a 2D field of view. And common examples of this are photomultiplier tubes or avalanche photodiodes, where essentially it's almost like you're counting the number of photons at each point. And these are commonly used in fluorescence microscopy. Now, multi-point detectors are probably something that all of you are much more familiar with because they're essentially just cameras. And these essentially are able to capture the entire field of view onto the sensor. And so there's no scanning involved. And depending on the technology, this may or may not be faster than single point detectors, but the important thing is that you don't have to have any mechanical scanning or motion to generate an image. And the commonly used cameras for scientific imaging are usually CMOS or CCD, and this basically just refers to the technology used for converting photons to electrons and then converting that to a voltage and storing it. We won't really get into the details of that, and you probably won't really need to get into too much detail. So with cameras, there's important considerations like the exposure time, and that again would depend on the quantum efficiency. Another really important consideration would also be the sizes of the elements in the camera or the pixel sizes and how you would sample this. And this is something we'll get into in more detail in the next session, but just remember that that's something you want to consider. Okay, so between Dylan and me, I think we've given you a fair sense of the different parts of an imaging system, starting with the imaging optics and focusing on the objective lens, to the illumination optics and the light sources, to finally the different detectors. So now it's important to see how all of these are put together in different types of systems and what you can do with them. So I'm going to give the floor over to Tara, who's going to tell you more about these interesting microscopy systems and their applications.
Thanks, Nish. So now that Dylan and Nish have given you a kind of an overview of the optics behind microscopes, we're ready to take a look at the systems themselves. So first, let's talk about what to keep in mind when choosing a microscope for your experiment. So as a very general perspective, you can think of it like this triangle with points for each desired parameter: high resolution, large imaging volume, and high speed. As you get closer to each point on the triangle, you get a little farther from all the others. There are similar trade-offs in microscopy. Generally speaking, the faster you want to image, the lower your resolution will have to be. For example, you may need to decrease your exposure time that Nish was talking about to get increased speed, which would lead to a lower signal-to-noise ratio and an effective lower resolution. Conversely, as we move towards the large imaging volume point of the triangle, the longer the imaging will take. So with this in mind, to choose the best microscope for a given experiment, you want to consider what the characteristic time and space dimensions of the sample are. So what's the smallest feature you need to see? What is your ideal imaging volume? And what is the fastest event you want to capture? This should give you an idea of where on this triangle you want to be and help you pick the best microscope accordingly. So here are the two main things I'm going to cover in this final part of today's session. My goal is just to provide a high-level overview of the types of contrast agents and systems that you can choose from and what their pros and cons are. So first, let's talk about contrast. There are two major types of contrast I'm going to cover, and the first is label-free or endogenous contrast. I will just focus on contrast based on refractive index mismatches in the sample. There are other types of label-free contrast. So if you've worked with cell culture of any kind, you've seen phase contrast images like the one on top here. And we'll talk a little bit about why this works. So the second is label-based or exogenous contrast. You've likely seen one or more of these types of contrast before. Broadly speaking, we can break this down into fluorescence or absorbance-based contrast agents. On the fluorescence side, there are two major types that I want to briefly distinguish. So in immunofluorescence, specific antibodies that target the proteins of interest are used, and they're called primary antibodies. Then a secondary antibody with a conjugated fluorescent molecule attaches to the primary. Immunofluorescence is typically used to stain fixed samples rather than for live cell or in vivo imaging because it requires the cell wall to be permeabilized for the antibody to enter. Another option is to use fluorescent proteins. For this type of labeling, the genetic sequence for the protein is inserted into the target so that it will be expressed. Fluorescent proteins are most commonly used for live cell or in vivo imaging. The famous example here is the GFP, or green fluorescent protein, which was derived from the jellyfish. So there are also absorbance-based dyes, such as those used for H&E and gram stains. And I won't be talking any more about this type of contrast, but feel free to ask questions at the end. So between these two major types of contrast, endogenous and exogenous, a wide range of targets and applications can be covered. So we're going to talk first about label-free contrast as it relates to refractive index. So in the last session, we discussed refractive index, which is the property that describes the speed of light while it's passing through a medium relative to the speed of light in a vacuum. So for example, the refractive index of air is very close to 1. So light is moving through air at almost the same speed as it would move through a vacuum. Water, on the other hand, has a refractive index of 1.3, so light slows down in water. The net result of this slowdown is that light waves will be shifted in space, and this shift is called a phase difference. Okay, so how can we use this fact to get contrast, say from cells? So consider the below scheme where light illuminates this adherent cell. Some light passes through only the glass cover slide. The glass slows down the light, so the wave is phase shifted. This will be our reference light. Some light passes through the edge of the cell, so it slows down not only when it goes through the glass, but also the cell, which is mostly water. A larger phase shift is added to this light, shown by the red area in the image by the wave. The sunlight passes through the center, thickest portion of the cell. Here it goes through glass, water, and also the dense nucleus that will slow the light further. The phase shift is largest here. So what does this all mean for what we see on the sensor or in the eyepieces? So to understand this, we need to remember the constructive and destructive interference properties of waves. So when two waves go through two different media with different refractive indices and slow down by different amounts, or if they travel a different overall path length, we see interference effects due to the resulting phase differences. So in the top image here, we see waves in phase where peaks overlap peaks and they add constructively. In the bottom image, we see waves 180 degrees out of phase where the troughs overlap peaks and they add destructively. Anything in between will give an intermediate intensity value on the camera. So the end result is an image like this one on the left. As you can see, there are lighter and darker areas of the image which represent constructive and destructive interference at the camera pixel. However, the contrast on that left image, which is called the Widefield image, isn't that good. To make the contrast better, you can add an additional component to the microscope. Oh, go back, wait. An additional component to the microscope called a phase ring. And so this will enhance the contrast from the interference. So you can see a sharper definition in the features with this type of contrast. So this very simple label-free approach is very useful. However, it doesn't give you molecular specificity. It tells you about the morphology, but not necessarily the chemical makeup. There are also label-free techniques to do this, and Nish or Paloma in the Robles lab could tell you more about them, but a highly popular approach to the problem of molecular specificity is to use fluorescence. So in our last session, which if you missed will be on our website, like Dylan said, we talked about how light carries energy. Higher wavelengths closer to the red end of the visible spectrum have less energy. You can see this schematically in the diagram here of energy levels. The arrows represent electrons transitioning from lower to higher energy states. The larger energy transitions can be made with blue light versus red light because the blue light carries more energy. If you ever need to remember the relative energy of different colors of light, just think of a rainbow. Energy decreases towards the red end. So we will use this energy carrying property of light to achieve the fluorescence effect. First, incoming light, referred to as excitation light, enters the sample. Next, the fluorophores absorb this energy and enter what is called an excited state. This state is unstable, so some energy is released as heat in a process called thermal relaxation, and the fluorophore moves to a lower energy state. Then, this energy is emitted from the fluorophore as a photon. The emitted light has less energy than the excitation light because of that thermal relaxation that occurred prior to emission, where some energy was lost, so it will be redshifted. Okay, so that's simple enough, but there's a little more to this story here. I've shown one possible way this energy state transition could go for a fluorophore, but in reality, every fluorophore will have a range of energies or wavelengths that will trigger excitation. This range is called the excitation spectrum, shown here as the blue curve. Likewise, there are a range of possible thermal relaxation processes and final emission energies, which make up the emission spectrum, shown in red. The y-axis essentially represents the probability of excitation or emission for a given wavelength in the spectrum. Okay, so why does this matter? When we image a fluorescent sample, we only want to see the fluorescence, not the excitation light. We want to block the blue and keep the red here. So how do we do this? So typically, this is accomplished with a set of filters in the microscope. These filters block some wavelengths and pass others. Here, a set of 3 filters works together, and their transmission is represented by the lines on this graph. So let's see how this looks in the microscope. So one is the excitation filter, called a bandpass here because it only passes a band of wavelengths. This could be used, for example, if you had a broadband or multicolor source like Nish talked about. But your fluorophore will only be excited by a narrow band of that source energy. You'll filter out all the wavelengths that you don't need, so you don't put a bunch of extra energy into the sample. You care about this because the additional light can excite other molecules that you aren't interested in, leading to damage in the sample called photodamage. So 2 is a dichroic beam splitter, which is a dichroic mirror in this case. Your dichroic mirror will pass some wavelengths and reflect others. You can see here that the incoming excitation beam and the returning emission beam will be able to follow the same path because between the mirror and the objective, but only the emitted light will be able to pass the dichroic mirror to go to the detector. The 3 is an emission filter that will only pass a narrow band of emission spectrum, eliminating any wavelengths you don't need, so you don't have any extra information on your camera. Okay, so that's one fluorophore, but what about multicolor imaging? So for multicolor images, multiple band passes will be needed to separate out each emission spectra. Here, CFP, YFP, and M-cherry are shown. These are fluorescent proteins. And there are multiple ways to accomplish this, and it will kind of depend on your experiment, what approach you choose. Here, a multi-band emission filter passes only one band from each spectra. So usually a few different sources and filter sets will be combined to accomplish this type of thing. So what do I mean by filter sets? What does this actually look like in a system? So in most standard commercial systems, a filter set or a filter cube will be a combination of an excitation filter, a dichroic mirror, and an emission filter, all in one little package. You can switch them out depending on what labels you're using. In an upright configuration, meaning the objectives come down from above the sample, the filter cubes will usually be found in a turret or slider above the objectives between the objectives and the eyepieces or detector. In an inverted system, they'll be flipped over because now the detector is on the other side, and they'll be found below the objectives. So if you need to design an experiment with multicolor labeling, it's worth checking the spectra of your source, like Nish talked about, and then also your fluorophores against each other to figure out what filters you'll need. So you can always reach out to the microscopy core in IBB for help with this. Okay, so now we're ready to look at a few different fluorescence microscopy systems. I'm going to cover 3 basic types of fluorescence microscopy that are the basis for many more advanced techniques. These are standard widefield, confocal, and light sheet microscopy. They're built around different types of illumination, which you can see here, and I will discuss more. So for widefield, as the name suggests, you're looking at the entire field of view in one shot. Here's a simple widefield illumination, and it uses a 4F lens system like we discussed and like Nish went over with Koehler Illumination to illuminate the entire sample with a collimated beam, shown here in blue. In green, we see the red-shifted emission light, which is collected from every point of the sample onto a camera detector. So when we look at fluorescence images taken with a widefield system, we can see they look increasingly fuzzy and blurry as we try to image thicker samples. And this is because that collimated beam illuminates the entire sample. So we will get out of focus light coming in from different depths, adding extra intensity to the camera without contributing any meaningful information. So additionally, this means that we're pumping extra energy into the sample, causing that photo damage that I talked about earlier. So overall, widefield is useful because it's fast, you can obtain high resolution, and it's as simple as it gets in terms of a setup. The downsides are it won't work well for thicker samples, and there's a large amount of photo damage away from the focal plane. So one way to address this first problem is to use computational methods like deconvolution microscopy. Deconvolution algorithms essentially remove out-of-focus light from each plane, leading to sharper images like the ones shown here. But this is computationally heavy, and it can produce artifacts. So what's another way to image thick samples? So for thicker samples, a popular alternative is confocal microscopy, or laser scanning confocal. So in confocal, instead of a collimated beam illuminating the entire sample, light is focused to a point. This focused point of light is scanned throughout the entire sample. However, with confocal, the magic really happens on the detection side, where the fluorescence is collected one point at a time. To eliminate any out-of-focus light before it reaches the detector, a pinhole is placed at the conjugate sample plane. And as Nish talked about earlier, anything in a conjugate plane with your image is going to overlap with the image. So the green lines here are light coming from deeper in the sample that will not be focused on the image plane. And we can see that they're rejected by the pinhole before the detector. So the point of light is recorded by a PMT detector, which is a single point detector. So when we compare the modalities, we see that confocal is winning in terms of sharper images. Additionally, because we can reject any light that doesn't come from the focal plane, confocal will be much better for imaging in 3D. The downsides are that it will be slower because scanning is required, and even though you're blocking out of focus light at the detector, there will still be that light going into the sample in other planes, so photo damage still occurs. So what if we want to keep the 3D benefits of confocal, but bring back the speed of widefield and reduce photo damage? So for this, there's light sheet microscopy. A light sheet microscope doesn't look like a normal microscope. In light sheet, the excitation and detection are completely decoupled. 2 different lenses are used entirely. The excitation beam comes in from the side, slicing the sample with a sheet of light. A second lens is positioned at a right angle to the sheet, so it sees the entire illuminated plane. This means that only one plane is illuminated at a time, so all of the energy being put into the sample is actually being used, which is not the case for confocal or typical widefield imaging. Additionally, because the entire field can be illuminated, this technique is fast. So we see that light sheet is fast, good for 3D, gentle on the sample. Why doesn't everyone use it? The answer is partially because light sheet is generally a little less user-friendly than confocal microscopy. For example, in many systems, the sample needs to be embedded in agarose, then dipped into the microscope from above, then rotated or translated to see different planes. There are so-called inverted light sheet systems, and that can accommodate a more traditional sample mounting. And overall applications are definitely increasing for light sheets. So you can expect to see this technique becoming more and more common. Okay, so now we've talked in detail about how to acquire good images, how a microscope works, but what do you do next? So please join us for our next session on image analysis, coming up next Wednesday, same time, same place. And now I'll pass it back to Dylan to start our Q&A.
All right. Thank you, Tara, once again. We appreciate you all for joining our second session of virtual series of Microscopy 101. Next session will be about microscopy image processing and analysis. Yes, so we look forward to seeing you all soon. Any questions you have, feel free to Yes, hands up button on your right side of panel or chat, or feel free to unmute yourself for questions. I will unmute everyone. Yes, question is not limiting to any content we discussed today, but hopefully free to all subjects.